This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 117, recorded on December 10th, 2015. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there.
How have you been?
Well, pretty well.
2 weeks it's been since we chatted, maybe longer. It's always nice to talk with you. Also joining me from Ann Arbor, Michigan, Michele Swanson.
Hello.
How have you been?
Great.
Is it really cold so you can't play golf there?
Well, believe it or not, it's 54 degrees Fahrenheit here today. But I actually have work to do, Vincent. I know you like to make the golf joke, but I haven't golfed in quite some time.
Really? I'm sorry.
That's okay.
I hope I didn't make you guilty so that
No, it just makes me wish that I could golf.
Yeah. Well, you have to go somewhere. Go to a meeting.
Well, Michigan is playing in a bowl game in Florida, so it just happens that I booked tickets last night.
Michele, I have a football story. But first, let me introduce Michael. Just a short one. Also joining us today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Welcome back.
Thank you.
It's been a long time since we had you on the show.
Yeah, well, you guys went off on your adventure to Michigan, left me stranded in South Carolina, and then I was traveling, doing a site visit at Oregon State.
Well, you're back. That's what counts.
Yep.
So, Michele, just very quickly, I went to visit Mississippi State last week.
Mm.
And you know, they're a football school. They have a big stadium. And when they have football games, everybody brings a cowbell and they ring it. Have you ever played Mississippi State?
No, we haven't.
Conference. So I gave a lectureship, which was named after someone. They gave me a plaque, and the plaque was actually a cowbell. And on the cowbell, they stuck the plaque, right? So they gave it to me in a little ceremony before my talk, and I was showing it to the audience, and they said, "Ring it, ring it."
That's great.
It was very cute.
Everybody has their own traditions, huh?
Cowbell, of all things. And, you know, on the ride from the airport, my host was saying, "Do you know about cowbells in football games?" I said, "No, why do I care about it?" He said, "Well, I have to explain this to you." I didn't know why at the time, but then later I found out.
Right.
Cowbells. So you guys in Michigan, you use horns or something?
No, we just pump our fist when we say hail.
Oh. Is the football season over? Or is it?
Well, the regular season is over, but we're going to a January 1 bowl game. Playing Florida at the Citrus Bowl in Orlando on January 1.
Are you going?
I am going.
Very good. Nice. Then you can play some golf because it should be warm there, right?
I'm hoping.
Good. Excellent. All right. Before we start with some papers, I want to read a follow-up email which has to do from our last episode, number 116. It's from Jessica, who writes, "I just finished listening to chewates and coconuts and was so elated to hear about the open access paper comparing soybean oil, beef tallow, and coconut oil effects on fungal colonization. I am a biologist by education, but I also have quite the crunchy side. If you have ever seen My Big Fat Greek Wedding, you may recall that the father figure uses Windex to solve all of life's problems, and that is me with coconut oil. I have several friends in different fields of science, and they poke fun at my infatuation." Pun totally intended.
Of course.
With coconut oil, and often demand scientific evidence for my claims. This is going to be a great addition to my arsenal, along with your entertaining and informative podcast. If you ever have the time or desire, you should devote an entire episode to coconut oil and all of its antimicrobial claims. Thank you for the segment and your weekly contributions to science. And Jessica is a laboratory technician.
That's great. Jessica, I hope you'll make that chocolate sauce.
Right. Nice recipe. We both got emails from Carney. Remember?
Yep.
I pronounce her name right because I know how to say
That's
Carney.
Jersey City.
Jersey City. This episode of TWiM is sponsored by ASM Microbe 2016. This is an all-new meeting that will take place in Boston, Massachusetts from June 16th through the 20th of 2016. It will feature the best microbial sciences in the world and explore the whole scope of microbiology. It's a mashup of the ASM General Meeting and ICAC, which used to be separate meetings, and now they're combined. And the result is a distinct forum for cross-disciplinary study, both in the classroom and in the integrated exhibit and poster hall. It's a unique meeting, and it's designed to spark collaboration and discussion. If your work touches the microbial sciences, this is the meeting you need to go to. Learn, connect, and advance at the first ASM Microbe 2016. Now, the abstract submission date is January 12th, so get going there. And where do you go to submit your abstracts? asm.org/microbe2016. All right, we have for your pleasure today a snippet and a full paper. And to give us a snippet of science, here is Michael Schmidt.
Well, thank you, Vincent. This is actually a compilation of two papers that appeared in back-to-back in Nature. The first that I'm going to devote the majority of the time is called The Complete Nitrification by Nitrospira Bacteria, and it was worked out by Holger Dahm's and Michael Wagner's groups at Division of Microbial Ecology and the Department of Microbiology and Ecosystem Science at the University of Vienna in Austria. And there's a large group of others from Russia, Germany, and other laboratories that have contributed to this really fine paper. And the second one, and they're both describing a really neat thing, and I'll get to that in a second, but the second paper is The Complete Nitrification by a Single Microorganism, and this is from a group in the Netherlands by Marty van Kessel, Dan Spieth, Mads Alberson, and the final author is Sebastian Luker. And they both worked on the complete nitrification looking for this enigmatic comammox bacterium. And so I have to give you a little background on this whole process of what nitrification is. And it's a component of the global cycling of nitrogen on the planet. And as all our listeners well know, nitrogen is a substantial component of the Earth's atmosphere, comprising 80%. And we cycle nitrogen, and the nitrogen that's used in all biological processes is in the form redox state of its most reduced form, which is ammonia. And so what nitrification is the conversion of ammonia all the way to its final oxidated state, which is nitrate. And for this process was first described by the great microbiologist Vinogradsky back at the turn of the 19th to the 20th century. And so here we are in the 21st century where this group from Austria and also the Netherlands is describing a single microbe that is able to take ammonia and oxidize it all the way to nitrate.
Can I ask you a question?
Sure.
So the reason you need to do that is so nitrates can be reutilized by organisms. Is that right? Because you can't use ammonium, right?
Right. We would have all these dead bodies piling up. We wouldn't be able to recycle. And of course, nitrogen fixation is a very energy intensive process. And if it goes off as gas, it takes a tremendous amount of energy in order to convert it. The other thing to appreciate is the form of fertilizer that we use in soils is typically anhydrous ammonia, and we put it down on our, we mix it with the fertilizer. We also use nitrate and nitrites in our fertilizer as well, playing on this whole nitrogen cycle to convert the oxidized to the reduced and the reduced to the oxidized, so that the plants can make effective use of the nitrogen that we're feeding them.
So when you fix nitrogen, when you fix gaseous atmospheric nitrogen, which is done by bacteria, the product of that is
Ammonia.
Ammonia.
NH3 or NH4 plus, depending upon which redox state it ultimately ends up in.
Okay.
But the oxidation state of the nitrogen atom is in its most reduced form. So nitrifying microbes, these are the microbes that are converting ammonia all the way out to the most oxidized form, nitrate, are generally perceived as specialized chemolithotrophs. And a chemolithotroph, literally converting that word to real meaning, is it's a rock eater. And the chemolithotroph obtains its energy for growth by oxidizing either ammonia or nitrite. And the canonical view of nitrification was folks always believed that this was a two-step process where ammonia, the most reduced form of nitrogen, was converted to nitrite, NO2. And it got enough energy from that process in order to grow. So that process generated about a negative 275 kilojoules to the organism in order for it to grow. It's literally using the ammonia as its energy source. And of course, since it's a chemolithoautotroph, it's able to fix CO2 in order to get its carbon. The second component of this reaction is taking nitrite and converting it to nitrate. And it doesn't get as much energy as you can well imagine because of the number of electron jumps. You're going from NO2 to NO3, or simply one pair of electrons, and it only gains a negative 74 kilojoules. But for many years, this is what puzzled energetic physiologists as they were trying to understand why a microbe didn't take ammonia and burn it all the way to nitrate. Why give away this energy source, nitrite, to another microbe? Because you can actually get more energy out of the process. You can get actually 349 kilojoules taking ammonia all the way to nitrate. So this group set out to see if they could find the comammox, where you have one common organism that is able to take ammonia and convert it to nitrite. And they used a variation of old microbiology techniques, and this is why I'm focusing on this paper, is they did an enrichment in which they used a defined medium in which ammonia was the only energy source in the plate, and it was a defined mineral medium. And again, they're looking for chemolithoautotrophs in order to select out the microbe that they want. And what they learned from this enrichment is they carried out this process for about 4 years by the time they went from start to finish. And there's some background that you have to know about the enzymology associated with these two fundamental processes, taking ammonia to the NO2 and then the NO2 to the NO3. And the enzymes were phylogenetically associated with different types of bacteria. So they were always cross-feeding one another, and they were growing in concert with one another. So what this group from Vienna did is, working with colleagues from Russia, they selected their starting enrichment from a water source that came out of a 1,200-meter well in the Caucasus of Russia that was drilling for oil. And this 1,200-meter deep oil exploration well enabled them to plant organisms that came out of this water source, and they enriched at 46 degrees centigrade to enrich for this moderately thermophilic. What they were hoping for is ammonia oxidating microbes. And they found a dense population of cells morphologically resembling Nitrospira species. And Nitrospira is the canonical organism that takes NO2 to NO3. But they couldn't find any organisms that typically look like the ammonia oxidizers that were in the same genus and species of the ammonia oxidizers. But they found a rod-shaped organism, and the as you can well imagine, growth at 46 degrees is pretty restrictive. So when they began to characterize and further enrich for things, they did traditional molecular biology, and they hunted with known bacterial or archaeal genes for the ammonia monooxidase subunit and the 16S ribosomal RNA genes for the ammonia oxidizer. And they found that they were absent from this enrichment. And the only way these organisms can grow is if they burn ammonia. So they were very puzzled by this, but they sequenced. And now in today's world, we have this ability to metagenomically sequence. And what they found is the metagenome of this fourth enrichment was dominated by two species. One was the nitrite oxidizer and a beta protobacterium that was affiliated with the hydrogen Filiaceae and two other rare populations. And remember that the hallmark of enrichment is to continue to enrich and to attempt to select out a dominant organism. And what they ultimately ended up enriching for is a population that was 60% of Nitrospira and 40% of this beta proteobacter. And
this took 4 years, apparently.
It did.
Wow.
And they took these sequences and they screened for key gene functions of autotrophic nitrification. And as expected, the Nitrospira coughed up the genes for NO2 to NO3. But more surprisingly is this Nitrospira also possessed homologs to the hallmark enzyme of ammonia oxidation. And here's where PCR primers may not always reveal the genes that are present, because they may just be too far off in order to get you a positive PCR. And so this ammonia oxidase gene that was in Nitrospira, which never before had anyone described having ammonia monooxidase, it had the three classic subunits, and it was genetically organized into the right operonic cluster. And they had two additional AMOC genes at other genomic loci, and they were dumbstruck at the genes are there. And so then they did the next experiment. They asked the question, could the pure culture of Nitrospira, and they named it Inopinata, which means unexpected. They were able to demonstrate by incubating this pure culture in mineral medium containing ammonia as a sole energy source and reductant.
Let me ask you a question, if I may.
Sure.
How did they get the pure culture? Was that just straightforward, just colony picking, or?
It took them 4 years through serial enrichments.
Oh, that's it. Right.
Serial enrichments in which they continue to isolate.
Why did it take so long? I don't understand. It was originally a small proportion of the sample.
That's the other piece. That's the other piece of their puzzle, because what their selection did, and this is common to both papers, is their selection relied on growth yield rather than growth rate. And so they were selecting for slower growers that generated more biomass rather than the other way around we do traditionally in microbiology, where we just select for lots of cells, quickly growing cells. And so that was the trick, is the slow growing. And there's a whole bunch of other enzymology that you have to understand. And for many years, and this was brought up in the discussion of their data, Costa and colleagues have made the argument that these enzymes would have to be somehow segregated in order for them to be optimal to do this ammonia oxidation, because Mother Nature selected this cross-feeding because of the efficiency. So clearly this organism is unique, and as Elio often says, stay tuned for further details. One of the neat things about ammonia oxidase is, of course, it's been used, the microbes responsible for ammonia oxidation have been used a lot by bioremediators, because you can feed it aromatic compounds and do all sorts of other magical things with these because they'll degrade recalcitrant aliphatic, aromatic, and halogenated molecules. So, you see these ammonia oxidators specifically cleaning up gas station sites and other things. And the fertilizer, if we had Ron Atlas on the phone, he would, of course, go on and on about the wonder of the ammonia oxidizers because of their importance for bioremediation. So their selection principally worked.
But let me ask you, I don't quite get it. Knowing what they know now, maybe in retrospect, isn't it possible to think of simpler ways than just looking for enrichment in liquid culture? Do these guys make colonies?
I believe they do make. They're typically growing in biofilms, and that's effectively the gist of the second paper. Is they were growing it in a biofilm, and I think this is the emerging area of trying to understand when microbes express certain genes is the state that they're growing in. And the hallmark of biofilms, of course, is their very slow growth rates. And it's this slow growth rate, but high yield, because the biofilms, of course, make big mats. And that's effectively, I think, the trick is they were literally selecting this out of probably the oil well, where it was growing on the aliphatics, aromatics, and halogenated molecules.
So you're saying that the reason they don't grow alone is because you can only propagate it as a biofilm?
Well, the second paper, yes. The second paper, yes. These, they were growing as planktonic cells.
So, Michael, you said that Mother Nature has separated these two steps. How cool would it be if this organism has both of the enzymes for both of the steps, but they segregate it within the biofilm, so spatially. So if they're in a pure biofilm, only one step of the reaction is done, in the center of the biofilm, and then the second step is done at the edge of the biofilm, something like that. Wouldn't that be cool?
It would be very cool. And there's precedent for that if we look at the blue-green algae with the heterocysts and how you fix nitrogen, where one cell fixes the nitrogen and the others deal with the oxygen that's being evolved, and they segregate it. And here's what the authors tumble towards the end of their paper, and they say, and I'm quoting, if nitrite is the only available electron donor, the gap in the reverse electron transport chain likely prevents nitrite reduction by the NRF-A gene protein and CO2 fixation by the reductive tricarboxylic acid cycle, which is the autotrophic pathway in this Nitrospira. And they present extended data in the supplement to this Nature article. Thus, this Nitrospira inopinata species may grow on nitrite only in the presence of a low potential electron donor for quinone reduction, such as hydrogen or intracellular storage compounds. So you're absolutely right, Michele, of thinking about this in a biofilm with these intracellular storage compounds. And so then they further contrast, and again quoting, in contrast, electrons derived from ammonia should be transferred to a quinone as the ammonia oxidase 1, AOB1, and thus enable autotrophic growth. And an ammonia transporter also enables this microbe to use ammonium as its nitrogen source, where they just take it straight as NH3 and put it into amino acids. And so I think this is really a interesting way of looking at this nitrification cycle, and it's so important in agriculture. I remember back when I was a freshman in college, and I wrote my first term paper for English Comp 101 on this particular topic about nitrification and fetal hemoglobinemia, where the nitrate was actually poisoning the infant. And so that's actually what got me into microbiology and had
the rest is history.
Well, it made me a nitrifying aficionado. I was fascinated by this chemistry, and to go to Vincent's earlier question, they have now since looked, now that they have better PCR for what this this comammox ammonia oxidizing gene cluster looks like, and they began to look for it, and it was overlooked in a lot of these soils because nitrification is found everywhere. It's found in dirt, soils, fresh water, seawater, deep sediments. Any place you look, you'll find nitrification going on. And they effectively conclude is that it was their selection process of looking for yield as opposed to cell growth rate.
And function rather than the gene sequence.
Yeah, functions rather than genes. So
I might date myself here, but if they had used southern blots and varied the stringency, would they have found it that way rather than PCR primer?
Which is why southern blots often are superior to PCR.
You get more information.
Well, I torture my poor graduate students asking them questions about stringency. And they said, where is that in the kit instructions?
Yeah, there you go. Kit.
Well, no, it's about, we all remember those horrible COT curves from qualifying exam days of, which is involved in stringency of how the DNA will bind to other DNA and how many of the interfering molecules you put in to change whether or not it lights up or not.
Michael, is it possible? So does this Nitrospira, what is it? Ima something?
Yes.
Inopinata.
Inopinata.
Does this exist anywhere else in the world, or did they create this by their 4-year selection?
No, they've screened elsewhere, Vincent. They've been able to pull it out of all sorts of places.
Okay.
So it's not an anomaly to this. And I think Nature's editors deserve great credit for publishing the second paper from the Netherlands on complete nitrification by a single microorganism. And when we get to the discussion about the water bears, I'll comment about how if you want to look at that, you can take a look at the methods they proved that the genes common for this comammox microbe and the careful controls that this group in the Netherlands took to demonstrate it was really one organism is what we need.
Wow.
Before we leave this, I want to comment on the word comammox.
Mm-hmm.
Comammox. It's derived from complete amino acid oxidizer.
Ammonia oxidation.
Oh, sorry, complete ammonia oxidizer.
Anaerobic ammonia oxidation.
Yeah, so it's like a unicorn, essentially. It was something that people had imagined might exist, but they had never found it. And they came up with this term, comammox.
And I put in the show notes, and this is in the open access, there's a review on nitrification that was published in the annual reviews of microbiology in 2001. So if you're interested in this, you can take a look at that annual review article, and that was by George Kalawachuk and John Stephan.
So, Michael, the second paper, can you put a link to that?
I will.
So that was also published in Nature.
Yeah, in the same issue.
Oh, so they had two papers on this by two different groups on the same.
Two different groups story. On the same thing that confirmed the same observation.
Very nice.
That's great.
Nice.
This is very, very exciting. And these are going to be useful, these bacteria, for remediation, right?
Oh, remediation. They're very important for agriculture because part of, rather than breeding pesticides that will inhibit things, we're going to probably begin to look at things that can control the nitrification process so that you can inhibit how much fertilizer washes away each year.
Nice.
Here's the stock tip for the day. Buy stock in Nitrospira.
Yes.
Ooh.
Does it have to grow at 46 degrees?
No, that was their trick to get it to grow. I mean, they're mesophiles, the thermophiles, they're probably psychrophiles. I'm sure at 1,200 meters, there was probably tremendous pressure, which translates into heat. And I think it was just their selection bias. And that's really, I think, the take-home message is designing a good selection, like anything, will help you get what you want. You just ask Mother Nature to provide, and she will.
But will they now, before we can apply it, say, in the farm industry, will we have to transfer this pathway to another bug that naturally grows in soil?
No, they were able to isolate this bug. Now that they got the gene sequence associated with this unique gene complex, this and the group from the Netherlands, I think we're going to begin to see this pop up in a lot of places.
And it could grow in soil.
Yeah.
It doesn't have to be in oil or.
No. It doesn't have to be in oil. I think that was a bonus in terms of carbon, because if you think about it, where was the microbe getting the carbon, the CO2, at 1,200 meters below the Earth's crust? It was eating the oil, the short chain aliphat.
Michael, I bet this really excited you, right?
Oh, yeah. That's why I sent it last week.
Yeah, it's really good. I appreciate it. Thanks very much. This episode is also sponsored by the ASM Biodefense and Emerging Diseases Research Meeting. Early bird registration rates are still on. This is going to happen until January 14th. After which time you have to pay more money to go to this great meeting. So register now and save. This is a meeting that takes place in Arlington, Virginia from February 8th through the 10th of 2016. It is the premier event to discuss how to defend against bioterrorism and shape the future biodefense research agenda. You can learn more and register at asm.org/biodefense. 2016, all one word. We thank ASM for their support of TWiM. All right, let's proceed to our paper, which is from Elio. Tell us about this one, Elio.
Well, first of all, the paper is called Wolbachia Co-infection in a Hybrid Zone, Discovery of Horizontal Gene Transfer from two Wolbachia supergroups into an Animal Genome. The authors are Funthouser Jones, Seinert, Martinez Rodriguez, Toribio Fernandez, Pita, Belia, and Seth Bordenstein. And this is a consortium of people from Spain and Vanderbilt University.
And France.
I have to thank Rami Aziz for pointing this out. Rami is a fan of ours. He listens to this podcast while he's driving in Cairo traffic. So we're doing him some good because that seems to be a problem there. So he is a microbiologist who teaches at the University of Cairo and has deep connections with San Diego. So thank you, Rami, for doing this. Anyhow, the paper is about horizontal gene transfer from bacteria and phages into an animal genome. And I thought that Vincent likes the subject so much that he should introduce it in general terms.
I love horizontal gene transfer, almost as much as vertical gene transfer. So vertical, of course.
What tangential? Is there a tangential gene transfer someplace?
Yeah. Vertical, of course, is the inheritance you get from your mother and father, but in horizontal is everything else. It doesn't come from your parents, but it can come from anywhere. And the issue is we are awash in DNA. DNA is everywhere. And I spoke to a scientist out in San Francisco years ago, Eric Delwart, who said DNA is everywhere. It's a real problem. And, and this DNA, which can come from many places, is taken up by both prokaryotes and eukaryotes and archaea. So horizontal gene transfer is very well known in prokaryotes. You know, it happens by phages, transduction, the uptake of DNA, and it confers new properties like antibiotic resistance. It also occurs in archaea and between bacteria and archaea. So the question is, what about eukaryotes? There it's been a little harder to pin down, and only recently when we have deep sequencing approaches are we beginning to see this going on. And there's even some evidence that the human genome has acquired genes from bacteria, although this remains to be proven. We've talked some examples on TWiM about transfer into eukaryotes. For example, on 98, we talked about genes that were acquired by ticks from bacteria that contribute to innate defenses of the tick. We even talked about possible transfers from bacteria into human cells. But in somatic cells, there's pretty good evidence. And there's some suggestion that bacterial DNA can even get into the germline of humans. We talked about horizontal transfer from bacterial symbionts into insects like mealybugs. And how does this happen? It could be naked DNA. It could be viruses. It could certainly happen between eukaryotes. But of course, there it's harder to pick up because we're all so similar. So if you could go from human to human horizontally, it would be really hard to pick up. That's why it's easier to pick up, bacteria into human as well. But we basically think that horizontal gene transfer has played an equal or maybe more important role in evolution than vertical transfer, because we're seeing more and more evidence of it everywhere in almost every.
Well, in bacteria, because bacteria have limited sex, this plays a huge role because it is, in fact, in an evolutionary sense, the mechanism like sex that enables recombinations and new forms of life.
Variation.
Yeah, but of course, since we have so many bacteria and archaea associated with us, the opportunities there to acquire genes from them, they're there for millions and millions of years. So,
right.
Okay, and as I said, it's not easy to pick up, but we're slowly getting better at it.
Well, and our body does have defenses for recognizing foreign DNA and trying to wrap it up and protect our genome from haywire.
And so do prokaryotes and archaea. And one of those defenses is called CRISPR, which is really famous these days, right? So there you go. Is that good, Elio?
Terrific. Now, today we are, as we record this podcast, a news item came out that was quite interesting. Let me background that. A report had been made that water bears, tardigrades, these are little guys who resist everything, desiccation, even in outer space, radiation, everything. And these guys who live in soils and waters and everywhere were claimed to have a whopping amount of foreign DNA from bacteria, 17% of the genome. And that claim is way beyond what anybody else has said. So it's very surprising. Well, today, a group in England, came out with a statement that this is not the case. This is not really true. This is contamination by bacteria. Well, of course, the authors of the original paper, who are in North Carolina, had thought they had taken care of this. They thought that they had taken into account the problem of bacterial contamination. Anyhow, right now, it's between these people. It's between the people who believe that there is, in fact, bacterial DNA in the genome of the tardigrades, as they're known, water bears, and the people who believe that this is, in fact, an artifact. So by the time you hear the actual podcast, maybe something else will have happened. But let me say that the folks in North Carolina answered the comment made and say, among other things, we plan to refrain from commenting more until we've done additional analysis that can shed more light on this issue, and we'll be happy to share what we learn between groups. And I think that's very reasonable. So, as Michael says, I am fond of saying, stay tuned.
Well, I think it's absolutely right. I mean, they had some really compelling evidence, and then this group from Edinburgh came out doing the same thing, and they're basically generating some controversy. So,
it's worth pointing out
they can both be right, can they?
Yes,
it's worth pointing out that when you do deep sequencing, it's very easy to pick up contaminants because it's so sensitive. And and you, Elio, and Michael and Michele, we worked in labs. We didn't even worry about DNA contaminating; it was everywhere. But nowadays, you have to be really careful, and that can get you.
Hey, this is slightly tangential, but how big are these tardigrades? These water bears?
I think they're about 1 millimeter. Are they? Who knows?
Let's see. I can look it up very quickly here.
The photograph is just so interesting. I mean, they're such creepy looking.
Fascinating beasts.
These are scanning EMs usually, so they look really good. The smallest ones are, so they can be 1.5 millimeters to less than 0.1 millimeters.
Wow.
That's right. That's right. There's some are really tiny. And 0.1 millimeters makes it. They have only about 1,000 cells. They're a little bit similar in a way to. You can think of them as, in terms of experimental work, similar to C. elegans nematode in terms of cells. All right. Anyhow, the tardigrades are going to be part of our lives, and so we might as well live with them. And they're fascinating. I mean, imagine they're the only thing known that survives in outer space experimentally. Think about that. Anyhow,
and since this episode's coming out after the new Star Wars movie will have hit the screens, it looks like one of the creatures that George Lucas could have imagined.
Right. Exactly. Okay, so now we have this paper on something totally different, namely grasshoppers. So this is a paper where I can give you the punchline first. Different kinds of grasshoppers, which are related, and I'll tell you how they are related. There's evidence in them for the horizontal transfer of 3 phage sets of genes, they call them haplotypes, as well as bacterial genes from the bacterium known as Wolbachia. So let me back up. What's special about this study is they study what's called a natural hybrid zone. I confess I didn't know the use of the term. A hybrid zone in nature is a place where on both sides of the hybrid zone are different species of related organisms, in this case, grasshoppers, could be some other insect. And in the hybrid zone, they actually mingle and hybridize. And for a population geneticist, this is heaven. This is really a great place to study what's going on because you can see the consequences of hybridization in action. So
plus, this was on the border between France and Spain. So the Pyrenees. So what a very heavenly for scientists.
Right. Well, it's very fun. It's fun. I mean, it's not surprising that a group from Spain was involved.
Right.
So they had studied this quite extensively. So this is the Meadow Grasshopper, and it comes in 2 varieties, Spanish and French. Plus, in the middle, they are hybrids. So this is a detailed study. I don't think we'll go into all the detail, but let's background this a little bit. First of all, we're dealing about microbial communities of many arthropod species, and these are very, very common. And most common is the genus, the bacterium Wolbachia, which is the most widespread among arthropods, apparently. It infects at least half all the known arthropods, as well as nematodes, which are invertebrates but not arthropods. And they do a lot of things to and for their hosts, including they do a thing called, they do a sex selection. They favor the emergence of females by a variety of mechanisms. I'll list them: feminization of genetic males, male killing, parthenogenesis, and a thing called cytoplasmic incompatibility. But let me introduce you a little bit to the grasshoppers because they're really fascinating. What's most fascinating about them is that they have about 10 to the 12 bases. Humans have 3 times 10 to the 9th. In other words, they're about 500 times.
Wow.
500 times more DNA. What this DNA is, I don't know. It's obviously a challenge even today to sequence something that humongous. Well, it's work. It simply takes more effort to do it.
It's fascinating, isn't it, that they would be so promiscuous, that they would take up so much mitochondrial DNA and.
Well, they take up, that's exactly right. They take up the rates of transfer from mitochondria to nuclear genome was faster or more than is normal. And this also suggests that they may take up other intracellular DNAs, intracytoplasmic DNAs like Wolbachia. So these guys seem to their genome seem to sop up DNA and not lose it. What they do with it, we don't know. Okay, but this is an experimentally interesting and different thing. So now, there has been there's quite a bit of evidence that Wolbachia genes make it into the genome of their host, and Wolbachia even target the germline stem cells. So this is a way to further their existence. So in the hybrid zone, they report that there are, as I say, 2 kinds of grasshoppers and hybrids in the middle. So the large amount of divergent supergroups of Wolbachia made their way into the host nuclear genomes, as well as the temperate phage WO, which is a Wolbachia phage, a temperate phage. They are known to be present in arthropods as well as in nematodes. And we're here to identify, as I said before, 3 haplotypes, sets of genes from phages. And when they set out to characterize the types, they found that the phage alleles are quite different. There's an allele called ORF7, which is found in multiple variations in many instances of horizontal gene transfer. So, and they go between hybrid and non-hybrid grasshopper genomes. 8 unique ORF7 alleles from 9 different individuals were found in a single hybrid population. So these alleles some of these alleles predate secondary hybridization. That is, they were there in the original genomes, in the genomes of the original grasshoppers, but others were found only in the hybrids. In other words, some of this happened before hybridization, some of this happened after hybridization. In a sense, it says anything goes before and after. It's not a function of hybridization, but it is hybridization fosters the transfer between organisms. So these events happened probably quite recently, and they're well preserved in the grasshopper genome because the grasshopper genome doesn't like to get rid of DNA. And some of these inserts are found in the same position of both kinds of grasshoppers. Others are subspecies specific. So again, the events occurred both before and after the divergence of the species and the subsequent hybridization. This is about it for the paper. The paper is more detailed than that, but it is a nice example of how you can combine studies of horizontal gene transfer with population genetics, essentially.
So you say these are relatively recent transfers. So what does that mean? How many years roughly?
9,000 generations they talk about, which is not much. I've forgotten how often. I'm sure that grasshoppers mate more than once a year, so it's a few years.
Yeah, okay.
Speaking of our fear of contamination now with being so reliant on molecular methods and PCR, I thought that the fish experiment where they did the hybridization and actually looked on intact chromosomes in the grasshopper and identified the Wolbachia genes was especially critical.
Yeah, you could do that with
Very good point.
You could do that with the tardigrades, right?
Mm-hmm.
That's right.
Yeah,
absolutely.
That would be a good one. Yeah.
That's interesting. Actually, when you think about it, what a simple thing that is. You want to find that the gene is in a genome, or a set of genes are in a genome. Well, look at the damn thing.
Yeah. Although that technique, FISH, this fluorescent microscopy, looking at hybridization directly to intact chromosomes, is not something everybody can do.
It's not
does
hard.
take
It's
some
not easy.
skill.
Yeah.
So I was able to talk with the first author, Lisa Funthouser Jones, and she just had a great conversation. She fell in love with genetics when she was in high school in Albuquerque, and in her lab class, they actually did crosses with parasitoid wasps, where like the mom had the female had one eye color, the male had a different eye color, and then they looked at the progeny and had to deduce what the mode of inheritance was. And it turns out in this particular species, the male, one of the parents is haploid, the other parent is diploid. And so it wasn't your classical Mendelian genetics. So she just fell in love with this kind of riddle solving and the power of genetics. So she pursued biology as a undergrad at Baylor. And in fact, in her sophomore year at Baylor, she was recruited to be a TA for a genetics lab. So while she was an undergraduate, she taught the genetics lab for 3 years of college, which really speaks to her intelligence and also her love of the field. She also did an honors thesis while she was at Baylor looking at fruit fly eye development. So she arrived in Seth Bordenstein's lab at Vanderbilt as a graduate student with a really solid foundation and a love of genetics. She said she really appreciates the group, that they are part of the ecology and environmental biology group at Vanderbilt, so it's interdisciplinary. Seth's lab also is half work on Wolbachia. The other half are interested in the impact of the microbiome on speciation, so another evolution question. But she also, I want to point out, you may have noticed on this paper, she's actually the corresponding author, not her thesis advisor, Seth. So that really indicates that she took the intellectual lead on this project, which was a collaboration established between the Spanish group and their group at Vanderbilt. And she also had the opportunity to mentor an undergraduate, Stephanie Seinhardt, who was the second author on the paper, who contributed for a number of her years as an undergraduate student there. So I will say that Lisa is pretty busy right now. She's set her defense date for her thesis as February 23rd. And as all graduate students out there can appreciate, she's got one more experiment that she's got to finish and write that one last big
always
paper.
is one more, isn't there?
Yeah, and so this paper that we're talking about was actually a side project that she and the undergraduate carried forward. Her main thesis project is on parasitoid wasps, and she's interested in particular on how these wasps can control the number of Wolbachia that are transferred in the egg cells, in the oocytes, to the next generation. So she has good evidence now that it's a maternally acquired trait, that the females control how many Wolbachia can contaminate, let's say, the oocytes in the next generation. So that's her big thesis project.
What's she going to do next?
She is considering a number of options. She's looking at postdocs, but she also really loves writing and having broad exposure to many different fields. So she's also considering working as an editor for one of our broad science journals.
When I was at Vanderbilt, I spoke with Seth Bordenstein. We have a TWiM on that. It's number 332. He talked a lot about Wolbachia and the phage WO in that episode. So
His
Elio
lab does remarkable work.
Elio, there's the
They do
DNA.
really remarkable work.
Elio, the Wolbachia DNA in the grasshopper. We have no idea if it's doing anything, right?
No, actually, they don't say anything about that. Yeah.
And
wait. No, that's not quite true. You got me there.
I think they had some
there
evidence.
is mention in the paper. I'd have to go back to the paper.
There was some evidence that there was enough genetic drift that they were no longer coding sequences. So it looked like, if anything, it's a small data set, but if anything, probably not.
Probably, but I'd have to reread the paper carefully. I'm sorry.
So we can use it as a tool, though, to trace what's going on in these hybrid zones. But in terms of function, there's nothing there.
Yeah, I asked Lisa about that, and she said there's no evidence for function.
Yeah.
Okay.
And yeah, especially these grasshoppers who take up so much DNA, it's probably just a crapshoot. Some is going to be functional and some not.
This is interesting. I didn't know that.
DNA hogs.
DNA hogs. I didn't know they did that. That's really interesting. Huh.
Well, they're part of the plagues of the Bible.
Yeah, they are.
And they have a term for it, which I loved: genome gigantism.
Yeah.
Wow. Genome gigantism.
Yep.
Oh, I like that.
And evidently it's true in this order of insects, the I'm not good at pronunciation, Orthoptera.
Yeah.
Orthoptera.
Sounds right.
Which are the crickets, the grasshoppers, locusts.
Nice. Oh, someone put a movie of a dancing water bear in there. That's nice.
I had to move it off my screen. It was so distracting.
That is so amazing.
It's really creepy.
But I think that would be good for Ray to put on the website, on the TWiM site, so folks know what the water bear looks like. And you'll see why I think it looks like a Star Wars character.
This is 1,000 cells. It's amazing.
Yeah.
Coordination.
It's incredible.
Yeah.
Wow.
Yeah.
Wow. All right. This is a wow episode. Let's read a couple of emails. First one is from Aaron, who writes, so this is for you, Michael Schmidt. Greetings. Thank you so much for your wonderful podcast. I'm a high school biology teacher, and I thoroughly enjoy your discussions of current articles and topics in microbiology. My background is in biochemistry, and your discussions are engaging and keep me thinking about the unseen microbial world that is all around us. I'm currently working on developing a curriculum where students will investigate the microbiome of Drosophila guts. I was fortunate enough to have a lab fellowship in an immunology lab at the University of Massachusetts Medical School this past summer, where I developed the fly dissection techniques and basic plating. As most of the microbiome of Drosophila is comprised of lactobacillus and acetobacter, I used MRS agar and ACE agar this summer, but I found the ACE agar difficult to work with, and I think it will be challenging for my students. Plus, it contains cyclohexamide, which I don't want to bring into my high school classroom. Could one of the panelists recommend some other potential media that I could consider for introducing these techniques to my students? Again, thank you for the wonderful show, and I look forward to future podcasts. I thought you might know this, Michael.
Well, the problem is the cyclohexamide is principally there to inhibit the eukaryotes that are coming along with the dissected Drosophila guts. So you can play the games with temperature, either going cold or hot, and see if that will work. And I don't know what the principal component of ACE is. I don't know if that's activated charcoal. I don't know what the acronym ACE stands for. And fortunately, my sound is still working, but my Google search at my institution isn't helping me with ACE.
If it's ACEs, that's a buffer.
No, ACE agar is,
that's
it
different.
looks like agarose digesting enzyme agar. So I've never heard of ACE agar before.
But in general, one of the challenges with studying the gut microbiome is that many of the microbes are not culturable. They live in these consortiums, and there's a kind of a food chain that they rely on. So that's why so much of the work done on microbiomes these days is molecular.
Right.
Well, it goes back to when you guys were at Michigan and you were talking to
Vince Young.
The other Vincent, and Elio brought up the chemostat that was based on stool.
Right.
And that was just absolutely fascinating. And that's what she's going to have to do is maybe set up a chemostat using the guts from the fruit flies and just constantly recycling in a little column the
but
microbes.
you still have a problem of detecting what's there. I mean, that doesn't go away.
No, but you'll at least be able to see if you can replicate it and figuring out who's there.
That's a bit, I don't know. That strikes me as a bit ambitious, you know.
Especially for undergraduates.
I mean, he just needs something that will grow some of the bacteria so that they can look at it. Maybe someone listening could give him some advice. We have lots of experts out there. Weigh in, please. You can send it to TWiM@microbe.tv, TWIM@microbe.tv. Our next one is from Ethan, who writes, Dear TWiM team, I have listened to TWiV for a while, but recently added TWiM to my repertoire, so forgive me if this has been addressed in earlier episodes. Having a background in chemistry and biology with an eye toward human therapeutics, if only in part for funding purposes, I was amused by the apparent surprise expressed by the hosts at a microbial secondary metabolite being used in human therapy. I would argue that the majority of small molecule therapies, especially antibiotics and anti-cancers to date, are derived from microbial secondary metabolites, either directly or some having been chemically tweaked for enhanced stability and pharmacokinetic properties. Chemists are getting better at rational design or the clunky approach of high-throughput screening of random chemical libraries to find new therapies, but 3.8 billion years of evolution has done a lot of work for us. We found this review paper on the subject, which is rather dated but seems comprehensive and doesn't appear to be behind a paywall. He sends a link to pharmaceutically active secondary metabolites of microorganisms. And
well, I'd kind of like to correct it a little bit.
Yep.
I don't think we were surprised. I think we were aware of what Ethan says, that many of the important compounds in the pharmacopoeia are secondary metabolites.
Right.
But the thing we were wondering about is what do they really do for the bug or for the organism that makes them?
Yeah.
And that's not always obvious.
That's right.
In fact, it's seldom obvious. So
And that's been the great
still,
debate.
from a point of view of the practicality of it, we're doing fine. But from the point of view of understanding the underlying reason why so many compounds are made by so many organisms, we draw a blank on that.
And how they contribute to fitness of the organism that makes
exactly,
it.
Exactly. That's not clear at all.
Yeah, I mean, for one of the examples we mentioned was lovastatin, the big anti-cholesterol drug of Merck. And, when you discover it, you want to get it into people, and figuring out what it does comes later, maybe. But yeah, I think you're right, Elio. We were aware of that, but I think we were just, we have wonder. We express wonder at these things sometimes, which may be taken for surprise, but it's not really.
But Ethan is absolutely right about the many microbial products that are now used clinically.
Always a joy to listen, he ends. The next one is from Mike. Hi, Professors TWiM. I am a recent convert to microbiology, and I'm preparing to take a medical microbiology course soon. In preparation, I have been reading several microbiology textbooks and getting a little frustrated. Every book starts with a long discussion of the marvels of the Gram stain, but yet after a few hours of Googling, I still can't find the answer to one very simple question. Are Gram positives and Gram negatives monophyletic? From my initial reading, it seems that Gram positives are a small monophyletic group, and that Gram negatives encompasses a massive group of bacteria so large as to be almost meaningless. Am I completely wrong? Do you know of good review papers that explain bacterial taxonomy? Right now, I am left wondering if our obsession with the distinction between Gram negative and positive is a relic of a pre-genomic age, or maybe I have no idea what I'm talking about. Likely. I would love to hear your take on this interesting question. I love all three of your science podcasts. I keep meaning to get to the Vertical Farm one. I've listened to almost every episode of the others. I love them all. Weather at Stanford. The weather at Stanford right now, 10 p.m. is 17 Celsius, 69% humidity, dew point 12 degrees with a cool breeze from the ocean. A lovely evening, although I really wish we would have some rain. Thank you for the wonderful podcast. They are all a fabulous service to the community and to the public. They are also wonderful, good fun.
Well, Mike has his facts absolutely straight. It's true that Gram negatives are as polyphyletic as they come. In other words, that there's no good way of handling their ancestry. Gram positives, I think, are much more bunched together, and certainly the Firmicutes are one big branch of Gram positives, but not all. So yeah, it's correct. And the Gram stain is still of great value in the clinical lab, because a lot is known about the Gram positiveness and Gram negativeness of bugs associated with the human body and disease. But otherwise, it has lost some of its glamour because there are too many exceptions to what seem to be hard and fast, simple rules.
But the other thing to remember is there are classes of antibiotics that specifically target Gram positives because of their thicker peptidoglycan layer, and there are antibiotics that target Gram negatives because of their outer membrane, and that's what folks have truly exploited. So looking at the development of new antibiotic targets, I think the Gram positives and the Gram negative cell structure, that wonderful differential stain enables us to think about. It constantly reminds us how the microbes are a very pleomorphic group of creatures that we can develop new specific targets based on what's there.
I found some articles which said that even the Gram positives are not really monophyletic. Right, they're as you suggested.
They're not totally, no. That's correct. But they're more so than Gram negatives.
Yeah, got it. Is there any
quantitative distinction?
Is there any source we can point, Mike, to? Is there an article? So in virology, we have a International Congress for the Classification of Viruses, International Committee for the Taxonomy of Viruses, ICTV. Is there such a thing for bacteria?
There used to be a journal on systematic taxonomy.
There is. There still is.
There still is.
Okay.
Oh, yeah.
And
Schmidt.
Yeah. I'd like to go back to the ASAGR. My internet finally woke up.
Sure.
ASAGR stands for Antibiotic Cell Extract Agar. And the recipe that I'm discovering on the internet is it looks like you can vary the antibiotic that's in it, and you just need one fundamentally that inhibits eukaryotes. And in this particular recipe, it's really a pretty simple agar that you could make. It has yeast extract at a final concentration of 0.1%. It then has yeast or E. coli cells, 0.5% dry weight, and then it has something called neomycin. And so yeast are relatively easy to get because it's Christmas time, so all your mothers and grandmothers and fathers and grandfathers are baking. And so, you can get packaged yeast that's already dehydrated. And so that's already your dry weight. So you can actually make up this agar. And so that may be a solution. And then neomycin at a concentration of 10 micrograms per mil, there are many topical antibiotics that are out in tube form that have neomycin as its active ingredient that's fairly inexpensive. So this may be something that you may wish to play with.
All right. Thank you, Michael.
Good.
All right, let's do one more. This one is right up Michelle's alley from Justin, who sends a link to a ProMed Mail article about the outbreak of Legionella disease in New York City, Legionnaire's disease, back in August, which Michele made fun of here.
No.
She said, you have Legionnaire's disease and we do not here in Michigan, right?
I said, I'm here in Ann Arbor where we don't have an outbreak.
That's
40
right.
miles to our east.
So Justin says, if I understand this correctly, so the article basically says that it was the source of the infection was cooling towers. They absolutely matched the strains they found in the cooling towers with the strains in the people. So Justin says, so if I understand this correctly, this means that the cooling towers spray a mist of the bacteria with or without amoeba into the air, and it rains down and contaminates the air around the building. This really conflicts with how some sources are saying it has to do with showers, since the cooling towers shouldn't feed into the showers at all. Could you provide a clarification for me as to what is actually going on? I'm inclined to trust the cooling tower mist idea just because of the source. So the source is ProMed Mail versus ABC News, but just that's a lot of bacteria you'd need to have raining down. So Michele, can you?
I'd be happy to. So what is in common between the cooling towers and the shower is that both of them can create aerosols. And so the reality is Legionella grows well in all kinds of water sources. It's in the built environment. And then if we have any type of man-made equipment that generates aerosols, that can be a source of infection. So the cooling towers we shouldn't use the term spray a mist because that sounds like it's part of the design of the cooling towers. Instead, think of the cooling towers as basically a pond that's elevated above the ground. And then when wind blows across, it can pick up moisture and distribute it. So that's the problem.
And then the organism could grow in the shower head as well, or in the pipes?
It can grow in the pipes, biofilms in the pipes. And then if you inhale water while you're showering and you have a really poor cell-mediated immune system, you could become ill.
Right. So Michele, I think you may remember the Hammer building, which is where I work.
Sure.
So almost every day I walk by, I feel a mist from on top of the roof.
Do you?
Because there are cooling towers up there.
Stop breathing, Vincent. Stop breathing.
No, just don't start smoking. You're probably fine.
And
most of us, in the typical outbreak, only about 5% of people become ill. The rest will have antibodies, show that they've been exposed. But Legionella, fortunately, is not a very potent human pathogen. Most of us can clear it without a problem.
There you go, Justin, right from the source, from the expert. Thank you,
Well,
Michele.
she's not the source of Legionella.
Oh, no. But she does have it in her laboratory, though.
I sure do.
Yeah. But she's the expert.
One of many.
Yes. There are lots of experts. But you're here with us, so thank you, Michele. And that will do it for TWiM 117. You can find it at iTunes and at microbeworld.org. And if you have questions or comments, please send them to twim@microbe.tv. Michele Swanson is at the University of Michigan. Thank you, Michele.
Thank you all. It's been a pleasure.
Elio Schaechter is at Small Things Considered. Thank you, Elio.
Oh, my pleasure.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
Good to have you back. We missed you.
I listened to the fun that you all had and was jealous.
I'm
We
Vincent.
do have fun.
We do. We have lots of fun. The Michigan thing was great because we had Michele, we had Elio, and then we had the daughter of Frederick Novi in the audience, which
That was
really
great.
awesome. Granddaughter.
Great-granddaughter.
Granddaughter. That's right.
Was it great?
Yeah, I
Yeah.
was sad that the camera was just fixed on the panel and it actually didn't rotate and show her.
Yeah, that's because it's my camera and I don't have anybody running it.
Yeah.
I don't have Ray Ortega or Chris Condayan, who, by the way, I thank. They are employees of ASM, and we thank them for their help. And I'm Vincent Racaniello. You can find me at virology.ws. The music you hear on TWiM is composed and performed by Ronald Jenkins. Check him out at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.